Thanks. Time as any, Mr. Newman. Take her down to periscope depth.
Yes, sir. Clear the bridge. Dive.
Stand right.
Let me see if I can, yeah. I'm here with Dr. Change Tan. She's author of the book Stairway to Life, and I just have a few, let me see if I can, yeah. I'm here with Dr. Change Tan. She's author of the book Stairway to Life, and I just have a few questions, so welcome. Dr. Tan, I wanted to ask you, how do you actually pronounce your first name in Chinese?
嫦娥.
Okay, I can't pronounce that. I wish I could speak Chinese.
So, well, yeah, it's the name the lady went to the moon with her rabbit in the Chinese legend.
Ah. So what sort of classes do you, I understand you're a university professor. What sort of classes do you teach there and elsewhere? And what sort of classes do you like teaching?
Mostly molecular biology.
Have you taught any chemistry classes?
In China, yes.
Yes.
I was trained as a chemist in China, actually. So I learned all my biology in America, which, well, actually, it's funny because there I cannot speak biology in Chinese.
You don't know the Chinese words, huh?
No.
Mm.
Are you planning to have a Chinese version of your book?
Yes, so a professor at Xiamen University, so a very famous university in China, she is translating the book, and she is done with the first part.
Just a cultural question. Do you think in China they might be a little bit more open to what you have to say in your book than, say, here in the United States or English-speaking countries?
I have no idea.
No idea. Okay. Can you tell the listeners what sort of readers would benefit from reading your book. I've read your book. There were parts I had to crawl through, which actually is kind of good. I learned a lot. But what sort of readers are you hoping to reach with your book?
I think anybody really is serious about what is life and where life came along, or how life came along.
What was your inspiration for writing the book?
May I share my screen to give you a long answer for this question?
Oh, please, please, share your screen.
Okay, let's see. I made a couple of slides for you.
Oh.
So, where did I go? Okay, so basically, you know, if I look back on my life, right, when I was born, where I went for my college education and graduate student education and then work, so I can basically separate my life into a period, for the most part, is a pig period and then a sheep period. So my whole life can be, you know, said in a few words, from a pig to a sheep. What happened was really, so if we look at myself, my bio sketch and my essence, right, and my science and my religion. So for most of my life, my science and my religion is opposing each other. Actually, I did not believe God for the most part of my life. And then I learned that I was double standard and God is love, but still I did not believe him until I realized in 2004 that really God is real and is living. So I decided to accept Jesus as my savior. But at that time, you know, my science and my religion, so initially it's opposing, and then when I became a Christian, they are separate. They are in two separate compartments. But now my science and my religion is harmonious, right? So what happens in between is the period between these two red dashed lines. So what happened there was in 2005, I started to work. You know, I came to University of Missouri in 2005, and in 2006, I started to teach molecular biology. So as most of people in China, I did not believe in God, and I thought life came from no life, and complicated life from simple life through mutation, natural selection was a long time. But in 2006, I started to teach molecular biology, and we talked about DNA replication, transcription, and translation, and how bacteria and eukaryotes do them, right? So here we're looking at the DNA replication initiation. For bacteria, we have an orange replication, which can be recognized by a protein called origin binding protein called the DNA. So it's the DNA ABC. And the binding will cause the melting of DNA duplex, and then the helicase and the helicase loader will join the team, and with recruiting of a couple of more proteins, then the replication can initiate. But when you look at what happens in eukaryote, right? So not only, many more proteins are involved. What is striking for me, or was striking for me, was that the molecules involved in eukaryote DNA replication initiation and those for bacterial DNA replication initiation are unrelated in the view of evolution. So they do not have a sequence similarity. They perform a similar function, but they are not related in any sequence, which is very, very striking. So at that time, I thought, well, that is very strange, right, for me, because I was teaching molecular biology myself. Teach myself to learn, right? Because I was trained as a chemist. I did not learn molecular biology myself. So when I needed to teach molecular biology, I need to learn it first. So it was striking for me, even though I was trained as a chemist and the little biology knowledge I have, I knew DNA replication is essential for life. And now we have two completely, different systems doing the same thing. Really, that suggests to me, it's impossible for life to evolve from bacterial to eukaryote. Right, because it's not like you can add a few things or change a few things. Then what functions in bacteria then can be modified or improved. So now we can have a eukaryote. We cannot say that, well, since this side cannot go to that, so we cannot get the complicated life from simple ones. And maybe we started with the complicated life. And then, life was streamlined or simplified, right? That would not work either, because what bacteria used were bacteria unique. They have no connection with what is used in eukaryote. So either direction is a dead end. It's really a dead end. So that was, a turning point for my life. I started to really think, where life came from, or how, right? How eukaryote arrive, and how bacteria were made. So in 2010, we have Craig Vendel. He said that they had created a first self-replicating species we have had on the planet, whose parent is a computer. And he actually embedded a quote in the bacteria they synthesize. He said, "What I cannot build, I cannot understand." So what they did was basically they took a bacteria called Mycoplasma mycoides, and they inserted a yeast vector to facilitate later on cloning processes, right? So they modify this genome and then isolate this genome, sequence it, and then added a few watermarks, including the quote I just told you, "What I cannot build, I do not understand." And then they started, they basically divide this genome into smaller and smaller pieces until like the pieces of DNA, single-stranded DNA, 65 to 100 base pairs. The whole genome is about 1 million base pair. So they started synthesizing the genome with oligos, which is synthesized in test tube, right? And then they assemble them into 1 kb segment, and assemble the 1 kb segment into 10 kb segments, and then 100 kb segments, and the whole genome. They actually assemble the genome and maintain the genome in yeast, a eukaryote. And then they isolate the genome and transfer the genome in a host genome, which is actually Mycoplasma capricolum, which is very similar to the genome donor to activate the genome. So the vendor team's cloning experiment, right? Yeah, eventually is they cloned the genome. All the functional elements were borrowed from pre-existing cells. So basically, it's a very big cloning experiment. But from their cloning experiment, really a lot of questions, related to Orange of Life were revealed, right? For example, they have made the whole genome in yeast, which is 1 million base pair, encoding the whole bacterial genome. But yeast, a eukaryote cell, it could not actually give birth a bacterial cell. It takes another bacterial cell, which is very, very similar, very, very similar to the genome donor to actually activate or to interpret and execute the instructions encoded in the bacterial genome and eventually make the synthetic cell. So their cloning experiment is very revealing about what really takes to make a self-replicating cell, right? From the synthesizing of the oligos, how can you put pure chemicals with the right, chirality, just link them together, right, to make a genome. The whole process is so revealing. So it's a very, very cool. I like that a lot. So basically, I started to thinking about life, understanding life by building it. That's how I'm teaching molecular biology class. I want to make my students really think. If we start from the beginning, right? If we start from the beginning, from the simple building blocks, what does it take to make a self-replicating cell? So in reality, I had this started with this. So if we started with basic building blocks of life, including amino acids, nucleobases, ribose, deoxyribose, phosphate, and the phospholipids, not even simpler one, right? So this is already the basic building blocks we can find in cells. If we start there, even, right, then we need to link them together, and then we find that we'll encounter the problem of homochirality and homolinkage, and later on is, even if we can link all the subunits together in the right way, is far away from getting a functional gene, a segment of DNA or protein that function as a gene that we see inside the cells, right? And then, you know, even if we can build one gene or a bunch of genes, you know, these genes has to be functioning interdependently and being, you know, tightly regulated. So even if we have a whole genome, like what the team did, right? They have a whole bacterial genome assembled in yeast. That genome is useless. That genome is useless until, you know, if you have a cognate or matching actually living cell cytoplasm and membrane, right? Then you can make a self-replicating bacterial cell. Giving bacterial, all the bacteria you want, right? You want to make a self-replicating eukaryotic cell is a whole different issue, very, very different issue. So if we look at the Orange of Life or life, we see really an exponential growing difficulties of steps. So maybe at the beginning we say, oh, well, maybe, you know, we can make amino acids, right? We can make amino acids from a simple mixture of methane, water, ammonia. You know, we can make amino acid, yes, but then when we, you know, try to go another step, I said, wow, you know, instead of we are getting closer to, you know, making a self-replicating cell, we meet a step which is even steeper, more difficult than the step we, you know, we thought we could solve, right? So basically every step you climb, actually no step has already been climbed, but assuming we could, right? So because now we are not climbing from the beginning to, you know, to each step, is we cannot climb any of those steps. But we assume, you know, because life already exists, so we assume the lowering steps are climbed, and then we start anew from, you know, from a different step, right? Then we find, wow, actually we are facing with a more difficult situation. So instead of like we are climbing to the, you know, the stairways to life, the further we go, the dimmer our hope become. Really, we become, right, more hopeless. Okay, the further we go, the further we find ourselves more hopeless. So initially, that's my understanding, and you know, my perception of life and the origin of life. So it's we see an exponential growing difficulties of the steps. Eventually, Rob, you know, and I collaborated, and so in that book, it became like this. So it's a little bit different than what I had initially, but that's okay, because actually we don't know how steep each step really is, because you know, humanly we cannot climb any of the steps, or naturally, you know, we cannot do that, all right? So we really do not know how steep the steps are. And that's my story. So
Wow. Thank you.
That was excellent.
Bravo.
Thank you for
Bravo.
sharing.
I have a couple geek questions, and we're gonna let you go because we know you're busy. And then when I get the video together, I'm gonna I'll also share it with you. So I'll get maybe pieces here and there.
That's
Because
okay.
I think a lot of the
And also when I describe, I could not find the right word, you know, because my language, right? So that was really very helpful from Rob. Rob is so good. Rob is so good at writing, and he is very good at asking questions. So really, he made me think a lot of times, and it's a blessing to work with him. Actually.
Yes, Rob Stadler was Change's co-author. How long did it take you to put together this book?
Well, the outline was started in 2012.
This is an 8-year project.
Yeah, so initially it's more like a textbook, and initially include two parts. The first part is the making of a self-replicating bacterial cell, and the second half is the making of a self-replicating eukaryote cells. And eventually I decided to separate the two topics to address Orange for Life first. And I met Rob, I cannot remember when, and I asked his feedbacks, and he gave me a lot of constructive feedback. So I said, oh, well, do you mind to be a co-author? And then he said, well, to be a co-author, I needed to contribute more substantially. So he started to take my stuff and scramble them, reorganize them. But at the end, really, the book is much better than what I could envision. So it's a blessing.